because they use usually exogenous proteins, which can be pretty difficult to express in a cell host, and can also be toxic for the host in particular. Also, those proteins can have very sensitive enzymatic activity, and so this creates many challenges for synthetic biologists to create viable cell host and high productivity. And so synthetic pathways can also create toxic intermediates or products. So to create, a solution that can help solve this problem is using a colocalization approach. And so by colocalizing the proteins, you can reduce the diffusion of the intermediates or the produce of this pathway. And so doing that means we can use synthetic protein scaffolds, which is actually the success varies according to the strategy used. And the strategy I'm interested in is the strategy of peptide-binding domains. And so peptide-binding domains, actually when you use your enzymes, exogenous proteins, and incorporate them into a host, you can also tag them with small peptides. And those peptides, because they are very small tag and not a big fusion proteins, it's less likely to affect the enzymatic activities of those proteins that you want to use in your pathway. And then you can co-express your enzymes with a scaffold, build on peptide binding domains. And those peptide binding domains, they're gonna bind to those peptides you tag your enzymes with. And so this is going to create a colocalization that can be very specific into your host organism. And so the peptide binding domains are small little units of proteins, and so they can also be expressed on their own, for example, to characterize their specificity and their affinity. And so specificity and affinity are the main characteristic of peptide binding domains. First, we can define affinity by the strength of the interaction between a peptide and the domain. And the specificity, we can define it by the number of peptides bound by a domain. So if the domain binds only one peptide, it's a very high specific domain. But if it binds also this peptide very strongly, it has a high specificity and high affinity property. And so I'm going to class the peptides into three categories. First, you have a wide range of affinity and a low specificity, which are very not the peptides and domains pairs I'm interested in. I'm more interested in the pairs that shows high specificity and high affinity, so I can create scaffolds with very predictable behavior. We can also be interested in the domains that are less affinities to the peptide, which can also be useful to create more modulable and maybe play around a bit more with the scaffold properties than high affinity ones. And so during my PhD, I'm interested in many questions. I'm just going to go through them quickly. And so the main question is, what are the determinants of the efficiency of a protein scaffold built with peptide binding domains? And so the first question is, how much individual peptide binding domains properties define the scaffold efficiency? And also, how does the host organisms interact with the exogenous scaffold? And then finally, if I have time to go there, I would very like to explore how much we can improve a scaffold with continuous directed evolution. So for today, I'm just going to focus on the first question, and also this specific part of the individual protein binding domains properties. And so the method we use a lot in the lab for a long time is the protein fragment complementation assay of the dihydrofolate reductase. And so basically, this method is happening in Saccharomyces cerevisiae, which is yeast, which is a model yeast used also to study evolution, but also synthetic biology. And so here we're going to use a protein fragment complementation assay to characterize the strength of a protein-protein interaction, which I'm going to use PPI for the rest of the presentation. And so in PCA DHFR, we study two proteins. So first the bait and then the prey. And those two proteins are tagged with two fragments of the DHFR. And so we split the whole protein DHFR into two fragments, and then we fuse those two fragments to our two proteins of interest. And so in a selective condition, the DHFR is an essential enzyme for the growth of the yeast. And so when the bait and the prey interact together in vivo, the DHFR can then fold into its complete structure and accomplish its function, which is essential again for the growth. And so the stronger the interaction between the bait and the prey is, the more DHFR, active DHFRs are present in the cell. And so with the increase of DHFR active in the cell, we can then observe more cell growth. And so here you can you have a very good relationship between the number of active DHFR in the cell and the cell growth. And so we can that way quantify PPI strength in vivo with this method. And so for the first part of my project was to test first if the PCA DHFR was also working well only with peptide binding domain expressed alone, and how can it be compared with experimental affinities or experimental KDs that were previously measured. And so here I'm going to start only with the two peptide binding domains. One AC3 domain, which bind polycolimoieties, like here, and there is also measurement of affinities already in the literature. We have also the same thing for the PDZ domain, one of the PDZ domain. And so those are the two domains I'm going to talk to you about today, mainly. And then the genetic construction I'm going to use to express those domains in Saccharomyces cerevisiae is a construction like this. So we're going to go through it step by step. First, you have two promoters, so the stick promoters, that express at the same level both of our peptide binding domains and the peptide. So internally, those two components are expressed at the same level. And then you have a fragment of the DHFR fused to the peptide binding domains and also fused to the peptide. So this is the way we're going to quantify the interaction strength. And this site terminator is in the middle of the construction and is able to terminate the transcription of both of our units in our system. And so to make sure that we don't also quantify too much, well, we want also to assess if we quantify a lot of noise using this system. So we're also using construction in which you only use peptide binding domains tied to one fragment of the DHFR, and then the DHFR fragment, the other fragment is expressed on its own, so without a peptide. And the same thing goes the other way around, when you express only one peptide, and in that way we're going to be able to assess the noise of our experiments. And so those constructions, they are inserted into a plasmid, And so the workflow goes like this. First, we transform all of our plasmid, which are individual combination of peptide domains or only peptides and only domains. We transform them in yeast. And then from those strains, we start preculture and triplicates, and then we draw them in our PCA selection conditions. And so when using this method, we also then put all the plates in a Tican optical density meter, and 3 times we're gonna see the growth of our yeast, and that way we can quantify our PCA DHFR signal. So this is the workflow, and the results I'm gonna show you here are mainly growth curves obtained with this method. And so the optical density is shown on the y-axis and the time is shown on the x-axis. And so this is the growth for the ACH3 domain. You can see that the ACH3 domains, with the ACH3 combined with the ACH3 motif and with the higher affinity, so 1 is the higher affinity, well, it grows very well in our condition. And then you can see motif 2 and 3, they grow a bit less, and motif 4 grow even lesser than 2 and 3. And you can see the empty motif where there is no motif and only the domain. It grows as well, but at least less than every other construction. And so this is a very positive finding for us that our assay of the PCA is able to detect changes in peptide domain affinity, especially ACH domains, which are very of interest in our lab. And then for the PBZ domain, we obtain also a very good growth with the first affinity motif, but then it gets a bit more complicated with the second one, which grows even less than without any motif. We're going to come back to that, but PBZ number motif 3 can also grow quite well above the empty plasmid. And so this is also good, but we're going to dive into why this can be happening a bit later. And so now that we know that the PCA is a good system to quantify the characteristic, the properties of our peptide binding domains, we wanted to see how much the fragments of the DHFR can affect also the experiment noise. So here we use this construction for the previous growth curves, but now we're going to also switch the DHFR fragments. So now the peptide is going to be fused to the fragment 1, 2, and the domain is going to be fused to fragment 3. So that way we can assess if the fragments also have an effect on our experiment. So this is another ACH3 domain, which is from a protein called DRB2. And so it's just another ACH3 that we tested. And so here you can see with the motif, with its motif with high affinity, it grows quite well. And with the MT also there is a bit of noise, but we're going to decorticate that. So here, on this graph, you have on the y-axis the derivative growth rate, which is the maximal growth rate in this curve. And so here you can see for the protein-protein interaction type with either fragment, it's pretty similar, so there is no significant difference. But for the negative control, so it's only the domain and not the peptide, you can see that the noise, the growth rates are different, which means probably that the one fragment is more likely to bind the fragment, the DHFR fragment alone can also bind or find the domain more easily than the other. And so this is going to be important for us to keep always doing the experiment with both type of construction all the time, so we can ensure that we get the most sensitive experiment. So yeah, the DH4 fragments can impact noise, which was expected because it does also in other type of experiments. And then we wanted to, so you might ask the question, why is it so noisy? Why is the empty construct growth also pretty well compared to the construction with the interaction domains inserted in them? And so DHFR, PCA DHFR always, not always, but it creates signal that is due to randomness, randoms encounters in the cell. And so because these random encounters happen, like the cells that express or always those two fragments, they are going to go at some point in time because these spurious interactions can happen. And so it was important to determine for us when this growth is happening, so later on we can concentrate our analysis on the first 25 hours, which the negative controls with only a motif and only a domain can, well, in the first 25 hours, they don't grow at all. So this was important for us to determine the time of the experiment we wanted to explore. So now we explored a lot of the PCA method. And so when we compare the PCA signal with the experimental data that was already acquired through in the literature, so we can observe with when you put the experimental kidneys on the log 10 scale and compared to the derivative growth rate, you can see a quite good correlation between the signal of the PCA-DHFR and what was obtained in vitro. So this is a nice observation. You can also observe here the outlier PDZ2 that is a bit concerning, but we're going to talk about it soon. And so I think that the correlation would be a bit better if I had tested more combination of domains and peptides. So it is a very good start, and we're gonna explore, keep testing more a combination of domains and peptide. And so finally talking about PDZ2. So what I did not mention earlier is that DHFR PCA is also dependent on protein abundance. And so if one of the prey or bait that you are testing is less abundant, then there is also going to be less DHFR fragments to do the fragment complementation assay. And so lower abundance in your proteins equals also less signal of the PCA-DHFR. And so what can cause this lower abundance of if the peptide is expressed at the same level in any of the construct that we did? Well, because those peptides can also be similar maybe at peptide degradation peptide present in yeast, well, this can maybe lead to an excessive degradation compared to some peptides. So it's very peptides or domain dependent, and also peptides, we don't know, it can also lead to instability into the construct. So maybe that's also why we saw an even lower signal for PDZ2 than the empty construct, because it was degraded a lot faster than the empty construct or other construct that we had. And so the way we're going to test this abundance question in the future is through an essay that was already previously published in 2014 using also PCA-DHFR. And so for this, we're going to only express construction of one fragment tagged with one peptide or one domain. And so this fragment is going to be cloned also in a plasmid. And this plasmid is going to be present in a strain which express the YFP tied with the other DH4 fragments. And so by expressive at high level the YFP, we are expecting to have random encounters in the yeast cell between our DH4 fragments 1, 2, and DH4 fragment 3, which allows growth. And this method was already used previously to have a very quite good correlation between endogenous proteins of yeast compared, when you compare PCA versus fluorescence measured by cytometry. And so this is the method we plan to use to measure also abundance of our peptide and domain. And so now that we have established that our PCA can measure binding affinity, can measure PPI strength, and can measure also abundance of the proteins, we want to test it also with a higher throughput. And so for this, we're going to design a library using all the known data about peptide binding domains. And so using this database of peptide recognition model, there is plenty of diversity of domains. There is mostly SH3 and PDZ domains, which we already showed worked pretty well with the PCA-DHFR. And so in this domain, in this database, they have measured, or they have resolved the specificity matrix of all of those domains. And so using this specificity matrix, it's not experimental dissociation constants, but it's a way to generate peptides with various levels of expected affinity. And so by modifying the maximal, the matrix specificity, we can change each amino acid and then obtain another score computed with this matrix. And so the higher the score is, the higher the affinity is expected to be. And so if you modify some of the important amino acid in this matrix, well, the score is going to be a lot lower. And then this can also project the affinity that we can obtain in our assay. And so using those kind of libraries that we can generate in silico, we can apply PCA-DHFR for binding and abundance assays using high throughput. And so here you can imagine that each color is a different sequence of domains or peptide. And so doing it for individual, a combination of maybe 1,000, 100 versus 100, so it creates a lot of combination. It's not very possible to do it one by one in a high throughput manner. So what we're going to do is we're going to use synthetic DNA synthesis to generate all of those sequences in one what we call a pool, which is one construction, one DNA pool, which very various DNA sequences in it, and so this also those O-pools can be then transformed into our plasmids, and then this plasmid can also be transformed in yeast for the final assay. And so here it's going to be a very similar approach doing the pool competition assay versus the one-by-one characterization. So first we create a preculture, which then we extract our cells for pellet extraction, for DNA extraction in the end. And then we apply our PCA selection, and then we extract our DNA to prepare libraries for sequencing. So what we expect to obtain in this workflow is high affinity pairs of peptide binding domains are going to grow a lot faster than the low affinity one with our assay. And so what we expect throughout the the experiment is that we start with different pairs of peptide and domains, start at the similar frequency in our library before selection, and then we start applying selection, and then the frequency of our pairs can differ when we consider their affinity. And so the variant frequency is going to change throughout the selection steps, which is going to be then be reflected in the sequencing data that we obtain at the end. And so using this approach, we can then map the peptide binding domain's property in the property space of specificity and affinity. And so mainly here, because we work with a high number of combination, we can assay specificity and affinity at the same time when a high affinity pair is going to be present here, but also if this same domain is interacting with various other peptides, well, it's going to also be in a lower specificity. If we can detect that this domain interacts also with many peptides, it's going to reduce the specificity variable. So here for the next step of developing the scaffold, we can also maybe focus on those high affinity, high specificity peptide domain pairs, or maybe also play around with the lower affinity domains, but still with high specificity, because we still want our scaffolds to be orthogonal and very specific. And so the next step, which is still pretty far away, so we're gonna, if you have any comments about this, I'll be happy to hear them. And so what I need still to do for creating the scaffold is first to find a reporter system to quantify the scaffolding efficiency. So if we combine two domains, well then we want still I'm going to get to that. So the reporter system needs to be composed of two components, because we want to start with small scaffold of two peptide binding units. And then we also want it to be small proteins that are colocalized by our scaffold, because it's going to be easier to express them and less problematic and results from small components. And also, we want to be able to connect the growth to the scaffolding efficiency for high throughput and sequencing. So if you have any idea, I would be very open to it. Right now, the model we think about testing is using again the VHFR activity for the scaffolding efficiency detection. And so here we have the scaffold composed of two peptide binding domains linked with a linker, and then we can fuse the two VHFR fragments into two different peptides. And so internally, those two peptides can be recognized by the same by the peptide binding domains. And so the two fragments can then colocalize and can then fold to get back the growth of the yeast. And so that would be option A for now for the scaffolding efficiency, but still open to suggestion. And so thank you for listening to me. And so I want also to thank all the team of the London Lab, and so especially Christian, which is allowing me a lot of freedom to explore the project I like, and then Alex, which is a major support in the lab for me. And so thank you. These are the references if you need. So thank you for your attention.
Thank you very much, Pascal, for your amazing presentation. From now on, the questions and discussion session is open, so feel free to ask your questions and comments. We will ask you to use the raise your hand function in Zoom or write your questions in the chat box. Congratulations. Yes, Professor Rama, you can go.
Thank you very much, Pascal, for accepting to present in the Synthetic Biology Club and for this very nice presentation. I have a couple of questions. It's a very interesting topic, in fact, which is talking about the scaffolding of providing scaffolds for enzymatic pathway. At least for our work, it would be very, very beneficial. My question is, now your target is having scaffolding system with high specificity and high affinity. This is your target, Pascal?
Yes, for this part of on my project, yes.
Yeah. Okay. So my question is, would you are you interested also to have a two-component system or link it two-component system or unlink two-component system? So I mean the scaffolding units, not the enzymes that will be attached to the scaffolding system. I mean the like SH3 domains and PTZ domains. Do you need them to be linked to each other and that these domains be in fact attached or linked to the target proteins or the enzymes that will be composing the enzymatic pathway? So the SH3 domain, and if we imagine an SH3 domain link it to a PTZ domain, so we have a two-component system. So these two components would have affinity to the enzymes like DHFR. Here, do you want to increase? So would you be dependent on relying on SH3 already linked to PTZ, for example, if we, or you will expect that SH3 and PTZ will have affinities to each other in your scaffolding system?
Yeah, I don't expect SH domains and PTZ domains to have affinity with each other because they are known peptide binding domains. They don't bind other globular domains. But if your question was about if you maybe worried about cross specificity between those two domains, well, those domains, they bind very different type of peptides. So maybe we can go back to well, they bind very different type of domains, but different type of peptides, and those peptides are going to be fused to the enzymes of the pathways. And so because we know that enzymes can be the activity can be sensitive to big tags or big fusion proteins, well, that's the idea why you fuse short peptides to them. And so the two components bind the two peptides binding domains that are linked together, which is the two-component system. Well, because it's an interaction, it's not a constant interaction. You can give a bit more freedom to the enzymes to have proper activities without being so much affected by a big tag of a domain. So I'm not sure if I answered the question, but
yeah, I got it. So if we refer back to the graphs, which in which you measured the growth with different pairs that you worked on, when you compare
this one,
these ones, yes, and the previous, yes, this one. So here, as we see that the empty plasmids were in fact not very different from the majority of combinations that you've tested. So
yeah.
Here your explanation is that there is a noise. Could you please re-explain that again?
Yeah, so PCA HFR is can be also noisy because it's when we express the HFR fragments in the same cell, well, just by chance or by randomness, they are going to encounter each other in the cell, the two fragments. And so because also they are expressed on the same construction, they are close when they are translated. So this also can increase the noise that we observe in the experiment, but still we can detect high affinity binding pairs that are the signal is higher than the empty plasmids. And so for the high throughput experiments that we are going to do, we know now that to reduce the noise, we're going to have to do the passages. So before 25 hours, so before the frequency of the empty plasmids increase in the pool.
Okay. So why if you use one of the two components of DHFR? I remember that you have F12 and F3. For example, if you express the F12 in the chromosome and just make the F3 from plasmid, so that then you have, constant number of copies, at least for one, because the copy number, yes, would also be a noise, but at least you have separate the two components and you can segregate the two components more efficiently. And
but all of that is because we want to do the high throughput sequencing afterwards, and so it's going to be very hard to link the information from the genome and the plasmids at the same time in the pool competition. And so that's why we wanted to use one construction, do it. But yeah, of course,
And also, I think also it could be interesting that you use inducible promoters for these proteins fragments, DHFR1, 2, and DHFR3, so that you can, for example, time control the abundance. In fact, because if they are if they are unconstitutive promoters, I think they will be expressed all the time, and their abundance will be very high to the extent that they will not detect. So they will be already affine to each other because they are very. So you see, when you have many people in the same room and they are already in affinity because they are in crowdness, so they are abundant. But you want to see their interaction that's active interaction, not passive interaction. To segregate active from passive interaction, you need to lower or control the abundance of your proteins. One mechanism is using inducible promoters, but the problem of that is that your colonies will not grow before you induce your promoters. But we can find a solution for that.
Actually, well, we could modify the promoters in the plasmid to put an inducible promoter. What we use a lot in the lab is a P-gal promoter induced with estradiol in the specific strains. So we use that a lot. And we are planning to use these inducible promoters to control the concentration of the expression of the scaffold when we are at this step. So the peptides and enzymes are going to be at stationary expression, but the scaffold is going to be expressed in different levels. So we can maybe that way control a bit more of the noise in the experiment.
I agree with that, but at that case, you need to lower also the number of copies of your proteins by expressing them, of your DHFR, by expressing them from the chromosome instead of plasmids, because plasmids are usually multiple copies, but when they are in single copy in the chromosome, this will make them less abundant relatively.
Well, the psych promoter we use is quite, it's not a strong promoter, but I agree that there is previous noise, like previous interaction in our strain, that's for sure.
Yeah. Thank you. These were my questions. Good luck.
Thank you. So does anyone have any more questions? I think no one has more questions. So this concludes our activity. I would like to thank everyone who joined and participated in this activity. And most importantly, thank you, Pascal, for your time and presentation.
Thank you for inviting me.
Thank you. As a quick reminder, if you want to present your work or an article, you can always register as a presenter on our website. Otherwise, you can register as an attendee. Also, today is the last presentation of the year, so the next presentation will be starting from January. Goodbye, everyone. Have a nice day and see you next year.
Bye.
Bye.
Thank you so much, Teresa.
Thank you, Professor. Bye.
Bye.